Tuberculosis (TB) is a communicable disease that is a major cause of ill health and one of the leading causes of death worldwide. About a quarter of the global population is estimated to have been infected with TB. TB prevention and control need effective and accurate screening and diagnosis product. I-SPOT.TB of Fosun Diagnostics is one product which is used for in vitro quantitative detection of Mycobacterium Tuberculosis specific T-cell immune response in human peripheral blood. I-SPOT.TB is designed based on interferon gamma release assay IGRA test. The Mycobacterium tuberculosis specific antigen stimulate effector T lymphocytes to secrete the cytokine interferon gamma, which is captured by the coating antibody and enzyme-labeled antibody. The spells are produced after adding the chromogenic substrate solution, and the number of these spells could be counted. One of the main advantages of the IGRA test for TB testing is that it does not cross-react with the Bacillus calmette-guerin BCG vaccine, which can cause false positive results with the tuberculosis skin test. I-SPOT.TB product is developed by Fosun Diagnostics. The main components are Mycobacterium tuberculosis specific mixed peptide antigen, phytoagglutinin PHA control, concentrated labeled antibodies, chromogenic substrate solution, and 1 microtiter plate, 96 wells. Supplied as 12 × 8 well strips in the frame. Coated with a monoclonal antibody to the cytokine interferon gamma, IFN gamma. At the beginning of the test, we should prepare the equipment and materials required, but not provided from the I-SPOT.TB kit, such as blood collection tubes, a centrifuge for preparation of PBMCs, the equipment and reagents to enable counting for PBMCs, either manually using trypan blue or other appropriate stain, and a hemocytometer on the microscope, a humidified incubator capable of 37 degrees centigrade with a 5% carbon dioxide supply, sterile PBS solution, sterile cell culture medium such as Gibico, AIMV, and RPMI 1640, pipette and tips. Specimen collection and preparation. Whole blood samples should be maintained between 18 degrees centigrade and 25 degrees centigrade until processed. Add an equal volume of sterile culture medium RPMI 1640 into whole blood and mix thoroughly. Then the blood sample was mixed with the lymphocyte separation solution. Take the tubes with mixed blood samples into a horizontal centrifuge and centrifuge at 1000 G at room temperature for 22 minutes. Collect the white, cloudy band peripheral blood mononuclear cells, PBMCs, using a pipette and transfer to a 50 ml canonical centrifuge tube. Bring the volume to 10 ml with cell culture medium RPMI 1640. Cell culture medium for the washing steps should be pre-warmed to 37 degrees centigrade before contact with PBMCs. Centrifuge at 600 G for 7 minutes. Discard the supernatant and resuspend the pellet in 10 ml fresh AIMV medium. After mixing with the pre-warmed AIMV medium, centrifuge at 350 G for 7 minutes. Cell counting. Carefully discard the supernatant. Add 0.7 ml of pre-warmed AIMV culture medium and mix well. Add 10 microliters to 40 microliters of 0.4% Triban blue solution. After mixing, take 10 microliters to count the cells, or use a blood cell counter to count directly. Plate setup and incubation. The I-SPOT.TB test requires 3 wells to be used for each patient sample. An active control and a positive control should be run with each individual sample. It is recommended that the samples be arranged on the plate as illustrated below. For the I-SPOT.TB kit, remove the pre-coated plates from the packaging and allow to equilibrate to room temperature. Add the samples and the controls. 1. Add 50 microliters AIMV cell culture medium to each negative control well. 2. Add 50 microliters specific mixed peptide antigen solution to each well required. 3. Add 50 microliters positive control solution to each cell functionality control well. To each of the 3 wells to be used for a patient sample, add 100 microliters of the patient's final cell suspension, containing 250,000 cells. Incubate the plate with the lid down in a humidified incubator at 37 degrees centigrade with 5% carbon dioxide for 16 to 20 hours. Do not allow the pipette tip to touch the membrane. Indentations in the membrane caused by pipette tips may cause artifacts in the wells. Manual plate washing. Take out the plate from the incubator, discard the liquid in each well, and pat dry. The reaction plate is buckled upside down on absorbent paper and patted dry. Add 200 microliters of PBS buffer to each well. Let it stand for 10 to 20 seconds. Wash and spring dry. Repeat 4 times. Labeled antibody incubation. Dilute concentrated labeled antibody in PBS to create the working solution. Add 50 microliters working labeled antibody solution to each well and incubate at 2 to 8 degrees centigrade for 1 hour. Spouse coloring and counting. Discard the labeled antibody and perform the PBS washes as described about 4 times. Add 50 microliters chromogenic substrate solution to each well. And incubate at room temperature for some minutes in dark. Wash the plate thoroughly with distilled or deionized water to stop the detection reaction. Allow the plate to dry by standing the plate in a well-ventilated area or in an oven at up to 37 degrees centigrade. Results interpretation. Results for the I-SPOT.TB test are interpreted by subtracting the spot count in an active control well from the spot count in each of the samples, according to the following algorithm. The test result is positive if the number of sample minus negative larger than 6 spots. The test result is negative if number of sample minus negative less than 5 spots. Borderline is the number of sample minus negative is 5 or 6 spots. Results is invalid when the spot number in blank is more than 10. It is recommended that the borderline and invalid results be retested with a new specimen.